Next, we are going to discuss other related techniques to PCR. And first one, it is the RAPD. RAPD means randomly amplified polymorphic DNA. Why the term random it is used here? इसकी रीज़न ये है कि जो DNA fragment हम amplify करते हैं by using PCR, they are amplified randomly. The reason is that here in RAPD, we are not going to use some sequence specific primers. इसकी क्या वजह है? इसकी वजह यह है कि जो टारगेट DNA है हमारा उसका सीक्वेंस हमें नोन नहीं होता. So unknown sequence it is the target. So that's why the primer that we are going to use, they are developed without any information of the target genome sequence. And they are mainly the synthetic oligonucleotides that are 8 to 12 nucleotides long. So they are short segments of oligonucleotides that are used as a primer. और इनकी जो लेंथ होती है वो 8 से लेके 12 बेस पेयर तक हो सकती है. When they are going to anneal within the DNA, so the annealing it is relatively random. So they can bind or anneal at several different locations within the genomic DNA, and then they are going to carry the amplification of the target genome. Because random amplification is there, so that's why the DNA sequences that show slight variation in their sequences, which is called as polymorphic DNA, it is being amplified, and the name to the technique is given as RAPD, that is random amplification of polymorphic DNA. This technique it has been developed several years ago, and it finds its application in order to find evolutionary or phylogenetic relationship among different species. तो इसको यूज़ किया जा सकता है हम कि जो इर्तिकाई अमल था जानदारों की डेवलपमेंट में, उनकी फॉर्मेशन में, वो कैसे हुआ. So that's why I told you that this technique that is RAPD, it can be used in order to find the phylogenetic or evolutionary relationships among different organisms. Either they are plants or animals or any other living forms. Now, this method, although it is relatively easy to perform because no prior information of the target sequence it is required. So what is the basic protocol here? How it can determine the phylogenetic relationship or relatedness among different organisms like some microbes. So here the next diagram, it is related to the relationship between different strains of Haemophilus. And here the first lane, it indicated the marker in different nucleotide lengths like kb, 2.5 kb, 1 or 0.5 kb. And these are different lanes on the upper side that represents two different microbial samples. And here the fragment pattern will indicate that whether these strains of microbes they are related to each other or not. For example, here at the lower side of the gel, number 1 pattern, it is the same in most of the cases. It indicates that here mainly 2 bands they are detected, here and here, and like in the on left side here and here. So mainly 2 bands they are detected or separated on the gel. So that's why we can say that these 2 bands they belongs to same species. So here different lanes indicates that they belongs to mainly same strain or microbial species. In the same way, the number 2 indicates that it belongs to same microbial species like here. And similar is the case with third strain. It indicated the presence of 3 bands at different locations. So in this way, by evaluating the pattern of the gel, we can say that whether the microbes they are similar with each other or not. So we can say that RAPD it can be used to amplify the DNA fragments more randomly by using a set of primer that may be 8 to 12 nucleotides in length, and the DNA fragments that are produced after amplification they can be separated on gel. और सेपरेशन के बाद इंडिकेट करेगा कि वेदर कि ये जो बैंडिंग पैटर्न है डीएनए फ्रैगमेंट्स का इट इज़ रिलेटेड विद ईच अदर आर नॉट. जैसा कि मैंने एक एग्जांपल आपको जेल की कोट की जिसमें जो सिमिलर बैंड पैटर्न है एम्पलीफिकेशन का वो ये इंडिकेट करता है कि वेदर माइक्रोब दे आर फाइलोजेनेटिकली आर एवोल्यूशनरी दे आर रिलेटेड आर नॉट. Although this technique, it is initially it was thought that it is a very good technique to find evolutionary relationship, but it has certain limitations. First limitation is that it cannot differentiate the allele that has been amplified. It is homozygous or it is heterozygous. Secondly, the basic reaction it is used, like the enzymatic condition, components of the PCR reaction, template DNA concentration, so it can also influence the quality of the PCR. And third, and the most important limitation is that RAPD, it cannot show the reproducibility of the results. So reproducibility, it is sometimes missing. Suppose we have amplified a DNA sample that have been extracted at a particular time from some living organism, and we have amplified it by using rapid PCR, and then we have observed the gel pattern. So we got a particular gel pattern at another time. Suppose from a same microbe we have isolated or extracted the DNA and again evaluated it using the same primer set by RAPD. So it is not second time necessary that the same bending pattern we will get as the first time. The reason may be that during the passage of time mutations may occur within the genome of that organism, so that target site for the primer node is missing. It is annealing at another location within the DNA, so a different bending pattern you can get next time. So that's why its sensitivity or reproducibility it is not sufficient in order to find evolutionary relationships.